Hello everybody, myself Sunil Pawar. Welcome you back for the new session of the 11th class biology subject. Okay, as you know that we are continuing the cytological process. So here today we are going to take the chapter 8, its second part, cell, the unit of life. Okay, as you know that in our previous chapter we talk about the cell as an autonomous unit, clear, cell as a cell contained unit, clear, and we also started that is the cell components and the cell structure, okay, in which we already learned about the cell membrane and the cell wall, right? So now here today we are going to start with the endoplasmic endomembrane system, clear. You know that this endomembrane system, which one thing, whatever the component in our previous discussion we talk about which one thing, that is the cell membrane and the cell wall, okay. So these are the outer component. Now we are going to discuss the endomembrane system, that means inside the cell, okay, whatever the protoplasmic contents are there, okay. So in this one, this different cell organelles we are talking here. So first of all we are going to discuss the endoplasmic reticulum, okay. As you know that the word itself is giving endoplasmic reticulum. Endo means inside, okay, plasmic means the plasma, that is the cytoplasm, and reticulum means reticulated structure, okay. So such reticulated structure which is present inside the cytoplasma is called as endoplasmic reticulum, okay. And basically we observe that it is a double membranous structure, clear, which is basically absent in the which one thing, that is the prokaryotic organism, okay. It is absent in the prokaryotic organism, and it is even absent in the human RBCs also, clear. So you know that exceptions are there, okay. You can find these exceptions anywhere. So similarly this human RBCs are also one of the exceptions where this endoplasmic reticulum is absent, clear. So basically on the basis of this position and location of this endoplasmic reticulum, we can it is responsible to divide this cell into two main components. One, that is the component, the portion inside the endoplasmic reticulum, and second the extraluminal, what you call, that is the cytoplasmic compartment or outside the endoplasmic reticulum, clear. So basically here this endoplasmic reticulum is simply divided into two types. One, that is the smooth endoplasmic reticulum, simply we call it the SER, and second is the rough endoplasmic reticulum, we call it the RER, clear. Smooth and rough endoplasmic reticulum, it is nothing special, okay. It is just because of the presence and absence of the ribosomes on their outer surface, clear. The endoplasmic reticulum, which basically consisting this ribosomal units on its outer surface, is called as rough endoplasmic reticulum, and the endoplasmic reticulum without this granular particulate structure is called as smooth endoplasmic reticulum, clear. Basically this endoplasmic reticular structures, okay, is in general, you can see that it about its origin, you can see it is basically started from the outer membrane, outer nuclear membrane, okay. It goes as a projection and reaches to the cell membrane, okay. So this tubulated structure, or when we want to discuss about it ultra structure, then it is basically three features. One, that is the cisternes, second is the vesicles, and third is the vacuolar features, or that you can say that the tubular, okay. Basically these vacuolar features are absent in the endoplasmic reticulum, but it is present in the Golgi bodies, clear. So here you can observe that third part, that is the tubulum, okay. So what is the first one? The cisternes. Cisternes are nothing special, it is just a double membranous parallelly arranged membrane tubule structure, okay. So we call it something cisternes. Second is the vesicles, okay. These vesicles are slightly projected portions on the peripheral portion of this cistern, that is called as the vesicles. Okay, it is believed that they are responsible for the production of the vacuoles and the lysosomic units in the next the cytoplasmic content. Clear? And the third one, that is the tubular. When this cistern is become branch, okay, and become tube like in nature, then it is called as the tubular structures. Clear? Okay, this is basic three components of this endoplasmic reticulum. And in general, if we talk about its function, then first of all, it provides the support to the colloidal cytoplasmic matrix. Clear? Someone is considered as it is as a this is the endoskeleton of the cell. Clear? Because it gives a basic support to the cytoplasm. It is responsible for the compartmentation. It is help in the rapid intracellular transport of the material. Clear? And this endoplasmic reticulum contains a variety of enzymes for the various metabolic processes. Likewise, this ATPase and phosphatases are there, which is responsible for the energy production especially. Clear? Now, here we are going to differentiate between the smooth and rough endoplasmic reticulum. If you observe this smooth endoplasmic reticulum, that is not associated with the ribosomal unit, while this rough endoplasmic reticulum is associated with the ribosomal unit. Its main function is synthesis of protein, while the main function of synthesis of lipids. Okay, it forms the vesicles and tubules, and cisternes and few tubules are there. Okay, it is usually found in the peripheral portion. It is basically found in the deep inside the cytoplasm. These are basic differences between the RER and SER. Okay, now we are moving to discuss the next cytoplasmic component, that is the Golgi apparatus. It is also considered as the Golgi body. One scientist named that the Camillo Golgi in 1898. He was observed that a densely stent structure, reticular structure, which is present around the nucleus, is called as Golgi bodies. Okay, and it is observed that it may have this disc-like space between 0.5 to 1 nanometer in diameter. Okay, and basically, when we talk about the occurrence, then this Golgi complex occurs in cell. Okay, but naturally it is absent in the prokaryote cell because it is a membranous structure. Okay, and just like that exception, in human RBCs it is also absent. Okay, in the sieve tubes of the plant cell, the phloem cells. Okay, now next one, that is the cisternes. Okay, so in this one, if we observe that this Golgi body is also composed of three parts. One, that is the cistern, second the vesicles, and third is the vacuoles. Okay, tubules are absent here. Clear? So these cisternes or the Golgi apparatus are stacked parallel to each other, and they vary in the number in a cell, and they often curve like a shallow bowl to give Golgi complex a definite polarity. Clear? So here is the one that is the cis phase, and second is the trans phase. Clear? So what is cis phase? This is the convex in shape that lies toward the cell membrane. And what is the trans phase? It is just the matured phase, and it is the concave in the shape towards the nucleus. Okay, so if you observe this diagram, suppose that here is the nucleus, then it is cis. This is the cis phase, concave, and this is the trans phase, convex in nature. That means it is toward the peripheral end, and this toward the nucleus end. Clear? And here you can see that there are the cisternes, this double membranous tubular structure which parallelly arranged here. Okay, and at the side you can see this vesicle, these vesicles are there, and this is the vacuoles which is released outside it. Okay, so in this Golgi complex you can see that these are basically secretory in nature. Okay, now just see the function. The Golgi apparatus is involved in the formation of the lysosomes, vesicles that contain the proteins and remains within the cell. Clear? It performs the function of the packaging the material. That means they are responsible for packaging, just like the formation of the food vacuoles. Okay, the air vacuoles. This is possible with the help of this Golgi bodies only. Okay, it acts as an important site for the formation of the glycoproteins and the glycolipids. As I told you, it is secretory in nature. I think so you remember the structure of the chromosome. Okay, on the apical most part of the chromosome, that means the acrosome, it consisted, which one thing, these Golgi bodies, and these Golgi bodies are responsible for the production of that proteolytic enzyme, which is at the time of the fertilization is responsible to penetrate the egg membrane, right? And now here the starch, carbohydrate, and these all are stored within this membrane structure. And next one thing, it also in the formation of the cell wall. As you know that during the cytokinesis, okay, when the plant cell is going to divide, okay, then during the cytokinesis, okay, so what happened, in the middle the two parallelly arranged layer is actually formed, okay, which further deposited with the help of this chitin, pectin, lignin, also this one thing. Okay, so these two parallelly arranged plates which form, which in this one, this Golgi bodies play such an important role. Now then we are going to move the third component, that is the lysosome. It is very clear from the name that is the lysosome. Lysis means to break down and soma means the body. That means such cell component which is responsible to for what? For the lysis process, for the breaking down. Okay, so these are also the membrane bonded structure, especially what type in nature? These are vesicular in nature, and it is produced by the Golgi apparatus. Okay, as I told you, Golgi body contain three components: cisternes, vesicles, and vacuoles. So these vacuoles get separated, then they are responsible to, this vesicles and vacuoles responsible to produce this lysosomes. Clear? And these are densely granulated structure which is basically containing which enzyme? Hydrolytic enzyme, and which is responsible for the intracellular as well as extracellular digestion. Okay, now then it is important for the which one, the internal extracellular breakdown of the substances. These are basically found in the animal cell. Okay, in general it is absent in the plant cell, but some lower plant is also consisting this lysosomic structures. Clear? It is freely suspended in the cytoplasm. In animal cell it is almost present, and except then these RBCs, human RBCs. Okay, and then here this the most abundant number in cells related to enzymatic reactions such as liver cell, pancreatic cell, kidney cell, spleen cell, leukocyte cell, and macrophages are there. Okay, you know that these all cells are responsible for engulfing, swallowing, and the destruction of the pathogens or the other components. Okay, so that's why these lysosomes are present in such a higher number in this one. Okay, then if you talk about the structure of the lysosome, that is nothing special. It is just simply surrounded by the plasma membrane, or what you call that, the cell membrane. Okay, and inside it this different enzymes are there which is basically secreted by this membrane. Okay, and its diameter is ranging from 0.2 to 0.5 micrometer in size. Okay, and basically they are having this hydrolytic enzyme, as I told you. Okay, and these enzymes are basically present in the crystalline form inside it. Clear? So these lysosomic enzymes are there. In fact, more than 40 varieties are present in the form of this enzyme, and on the basis of their activity, or on the basis of their nature, we categorize them as a proteases. That means the group of the enzyme which is responsible for the digestion of the protein. Then the lipases, the group of enzyme which is responsible for the digestion of the lipids and the fatty fats. Okay, amylase which work with the carbohydrates. Clear? Nucleases which works on the nucleic acid, that is the DNA and RNA, and this phosphoric acid. Okay, so remember these all are basically hydrolytic in nature. That means they work with the digestion by hydrolyzing the substances, okay, and mostly the lysosomal enzyme function as the acidic medium. So remember, this acidic medium is basically required, and then on the basis of the origin we can categorize these lysosomes into primary and secondary lysosome. Okay, what is the primary lysosome? Such a small sac-like structure, okay, which is directly originated from the Golgi bodies, okay, which is enclosing this enzyme, which is basically having storage granules or the storing enzyme inside it, that is called as the primary lysosome. Then secondary lysosome, it is basically formed by the fusion of the primary lysosome, and they are basically having the material and the enzymes are there, okay, and these materials are further progressively digested inside this one thing. So in the different secondary lysosomes you can observe, okay, even some residual bodies are also there in the form of lysosomic. When this undigested unbreakable substances remain, then it is called as the which one thing, that is the residual lysosomal structure, and someone is also excretory in nature, okay, who is finally responsible to hold only that waste material or the harmful material which we need to excrete out, okay. So this cellular component is carried out to the side, okay. So this is the function. They digest the food content, that is the intracellular digestion. They also perform the extracellular digestion by releasing out this enzyme outside the cell, and they also digest the old and the useless organelles of the cell, and they also have the functioning in the cell division too, okay. So these are the called suicidal bags due to the presence of the hydrolytic enzyme. Okay, D. Dewey, he observed the rounded bodies in liver cells and called them the pericanicular dense bodies. Okay, remember, the lysosome is also called the suicidal bag. Why it is called it? Because in some specific condition, like with the extreme starvation, okay, when cell couldn't find any kind of food material for their growth and development, okay, so in this case these lysosomic units are responsible to engulfing, okay, and they start to engulf the small cell components, and by digesting them they help to survive these cells, okay, remember. Now we are moving towards the next cell organelle, that is the vacuoles. These vacuoles are the large membranous sac found in the cytoplasm, and these stored substances that are not essentially useful for the cell, likewise water, sap, excretory products and the other materials too, okay. Now these plant vacuoles contain not only the water, sugars and the salt, but also contain the pigments and the toxic molecules, and also occupy up to 90% of the volume of the cell, right. Now these vacuoles are responsible for the concentration of the materials tend to the higher in the vacuole and then to in the cytoplasm, okay. These animal cells are having less number of vacuoles, but these are very prominently and in high number present in which one thing, that is the plant cell. You know that such a big air vacuole is present where in the plant cell, who is responsible to shift that specific position of the nucleus at the peripheral side, clear. So here these vacuoles can be divided into two. One under the contractile vacuole and second the food vacuole, okay. These contractile vacuoles are basically responsible to collect and separate out this excretory material, okay, while these food vacuoles are responsible to hold and store the food material, which is further responsible for the digestion these purposes, okay. Now we are moving forward. One of the most important cell organelle, that is the mitochondria, clear. It is also a membrane bonded structure, okay, which is the essential component of the aerobic respiration in eukaryotes especially, and because it is responsible for the energy production, so we call it the which one thing, powerhouse of the cell, okay. You know that there are two processes in the aerobic respiration. One, that is the glycolysis, and second is the Krebs cycle, and third is the electron transport system. Clear? This glycolysis is generally observed where in the cytoplasm, while this Krebs cycle and electron transport system is observed where inside the mitochondria. Okay, so that's why it is called as the powerhouse of the cell. Okay, its occurrence is evenly present in the plant cell and the animal cell, and especially it is present in higher number where the anaerobic respiration takes place. Okay, and its shape and size, if we observe, then the shape is quite varying in nature, from granular fibular shape to spherical, oval, discoidal. Okay, and even you can observe the different shapes too. It's a normal size, average from 2 to 6 micrometer in length and 0.5 micrometer in diameter. Clear? Basically this one, its diameter is in generally ranging 0.2 to 1 micrometer in size. Clear? Now if we observe its that is the electron microscopic structure, then you can see that it has double membrane structure, the outer membrane and the inner membrane. Clear? But if you observe, then this inner membrane is become it shows the interdigitation, which is responsible to increase the surface area, and this projected units are called as which one thing? It is called as the cristae. Okay, and these cristae are having a particulate structure which is present on this membrane, that is called as F1 particle, and this F1 particle is basically responsible for the ATP's production. Clear? In this matrix or this fluid, they are also containing DNA also. Clear? They have their own DNA molecule which is responsible for the production of this large number of enzymes. Clear? And this is called as the extra chromosomal DNA. Clear? You know that this extra chromosomal DNA is only present in the mitochondria as well as the chloroplast, except then the nucleus. Clear? And here this different type of enzymes are present inside it, which is basically responsible for oxidative oxidation of the food material. Clear? And here is basically these ribosomes are also present. These are in general 70S ribosomes are there, which is responsible for the production of this one thing. Clear? So basically mitochondria are responsible for the intermediate, for the synthesis of the several biochemicals like the pyrimidines and the alkaloids, and the most important function, that is the production of the energy. Clear? They are also responsible for the production of the enzyme which is helpful in the synthesis of the fatty acids. It also helps in the regulation of the cellular metabolism. They also helps in the apoptosis, that means the program of the cell, that means the gradual reduction in the number. Okay, and the gradual reduction in the production of the energy is called as the programmed cell death. Clear? And each membrane, it also shows the membrane potential. Okay, and it is considered the second most largest cell organism which is present in the cell. Okay, now here we are moving toward the plastids. Basically these plastids are absent in the animal cell, but they are present in the plant cell. Clear? It is also the double membrane structure. Okay, and they are having their own genetic material, specifically DNA, and they are also quite bigger in size, so even they can seen with the help of the simple compound microscope too. Okay, so basically these are present in the plant cell, and even in the euglenoid cells are also there. Now you know that these euglenids are considered as the connecting link between the animal and the plant cell. Clear? Now when we talk about their types, then they can be basically differentiated into three different types. Okay, one is the leukocyte. Leukose means white, that means such plastids which is lacking any color. So simply colorless plastids are called as leucoplastids or the leucoplast, and they are basically responsible for the storage of the different material. Clear? So on the basis of their storage, we can categorize them in the amyloplast, aleoplast and the aleuroplast. Amyloplasts are responsible for the storage of the carbohydrate, okay. Then this aleoplast is responsible for the storage of the oil and fats, while this aleuroplast is responsible for the storing of the which one thing, that is the proteinous contents inside it. Clear? Now then here the second type of plasts are there, chromoplast. Chroma means the color, okay. Such plastids which is having colorful pigment, that is called as chromoplast. Simply you can see that this yellow color, reddish color, it is because of only the presence of this colorful pigments in this plastids, okay. Simply this carotenoids are also there, okay. This xanthophyll is also there, which is giving a specific pigment to the orange and this kind of thing. Clear? Now then the chloroplast, finally you know that the cell organelle which is specifically present in the plant, which is responsible to give them the green color, and this green color is because of the presence of the chlorophyll inside it. Clear? It is responsible for the photosynthetic activities of the cell, okay, which can be only performed in the presence of the sunlight, and that's why it is very important for the plant and the whole living world also. Clear? This chloroplast is basically occurs in all the green cells, specifically the mesophyll cell, okay, or you can also call it the collenchymatous cells, which is basically responsible for the fixation of the sunlight in the form of the chemical energy. Clear? It may be discoidal, it may be ribbon shaped, it may be oval, okay, and it basically having the length of 5 to 10 micrometer, then its width is 2 to 4 micrometer in nature. The numbers is also varies from cell to cell. Just like in a chlamydomonas, a single cell structure, 2 to 40 per cell is present in a mesophilic cell. Clear? If you observe their ultra structure, okay, then you can see that it is also double membranous nature. It is simply plano-concave convex in nature, okay. One side is quite plain in nature, while the second side is the convex in nature, okay. In this one, in the inner one, you can see that matrix is filled inside it, where this stake-like structure is observed, which is in generally called as granum. This granum is composed of this thylakoids. This thylakoid is especially the double membranous structure, okay, which is containing which one thing, chlorophyll, that is the pigment inside it, and when they are in the form of a stake, that is called as the granum. Clear? This all granum attached with the help of this what do you say, that is the intracellular network, okay, and it remains dipped in this matrix which is called as the stroma, and whatever the enzymes which require for the photosynthetic process is present in the stroma, okay, and further the solar energy is trapped and finally converted into the chemical energy. Clear? Remember this one thing, or the function of the chloroplast. If you observe that, then first of all it is helpful in the photosynthesis, okay, just for the trapping of the solar energy and storing them in the form of the organic food material, okay. It consumes the carbon dioxide and release of the oxygen gas. Clear? It may be changed into the chromoplast in order to provide the color to many flowers and fruits, which is helpful for the pollination and all these purposes. It is also responsible for storing the fats and lipids, okay. Its function is also transduction of the energy too. Clear? The sum total of all plastid cell is called the plastidome, okay. Remember this one thing, this term plastidome is which one thing, the all total sum of the plastid, whether it is leucoplast or chromoplast or the chloroplast, okay. And the chloroplast with nitrogen fixing genes are called as the nitroplast, right? And the space between the two membrane is called the intramembranous space, okay, which is basically specialized in the different purposes. These ribosomes are present, which is one of the 70S and 80S unit, which is in this, okay. And even This chloroplast is also considered as the self-replicative unit. Okay, after the division of the uneven numbers in the two plant cell, it recover the number. We know that this ribosome is basically made up of these two units. This, you can see that the 70S ribosomal unit is basically the characteristic feature of the prokaryotes, and this 80S is present in the eukaryotic cell. So this 70S is made up of these two small units, that is the 50 and 30, while this 80S is made up of 60 and 40. And we know that what is the basic function of the ribosome? Ribosome is basically responsible for the protein biosynthesis. It is also termed as the protein synthesis factory. Clear? Now here we are going to the next one. Clear? Now what is this, the next one? That is the cytoskeleton. Clear? In this cytoskeleton, if we observe, then basically the cell component which is responsible for giving the internal support to the cell, that is called as cytoskeleton. And basically this is the network of the this proteinous filaments and the tubules, okay, which extend from nucleus to the cell membrane, and it is in general responsible to give a stability and a primary strength to the cytoplasm and the cell. Clear? Now if we observe, this is only the characteristic feature of the eukaryotes, it is totally absent in the prokaryotic cell. Okay, if you observe the function, then in general it is responsible to maintain the shape and size of the cell. Okay, it is also involved in the many functions in a cell, as well as the mechanical support, right. So this is just the single function of this one thing, the mechanical support and the shape and size. Okay, now we are moving toward the next cell component, that is the cilia and the flagella. As you know that this is the characteristic feature of the phylum Ciliata and the flagellata. Okay, these are basically responsible for the movement of the cell, and even in some cases the locomotion of the cell. And if these cells are present in the interior portion of the body or the organ that is responsible for the this kind of flagellar movement, and also this one thing, right. I think so you are quite familiar with the structure of the euglena and the paramecium. Okay, in the paramecium you can see that its whole body surface is covered with the cilia, while in the flagella you saw the euglena in eukaryotes and the bacterial cell in the prokaryotes. Okay, where these flagellas are quite longer in nature, they are very few, they are not uniformly distributed on the body, they are located on a specific position on the body. Okay, while this, and they have their independent movement, while if you observe the cilia, then they are having a synchronized movement of the body, and they are basically not only responsible for the locomotion, but they are also responsible for guided the transportation of fluid. Just like that, they are present on the internal surface of the ureter, they are present in the fallopian tube. Okay, so even so the ciliary epithelium is also which is responsible, sometimes sensory in nature, to having the cells. Clear? And if you observe, then basically in this flagella you can see that there is basically if you observe that I think so you remember when we talk about the prokaryote and eukaryote, then we learn that there is a two type of arrangement, 9+2 and 9+0. Okay, so here are these microtubules are there, okay, which is arranged in the form of the group of 3 perpendicularly. Clear? So when there are 9 arranged in the peripheral portion and 2 arranged in the center, then this arrangement is called as 9+2. And if this center 2 microtubular groups are absent, that is called as 9+0. 9+0 is the characteristics of the prokaryotic, and 9+2 is the characteristics of eukaryotic one. Okay, and we know that this bacteria and this one. This bacteria are especially having both these two, flagella as well as cilia, while the eukaryote is having either of these two one. Okay, if we come to discuss the differences, then cilia are short in size, as well as they are 22 to 20 micrometer in length, while the flagella are long, pipe-like organelles and 10 to 200 micrometer long. Okay, these are present relatively in large number while they are very fewer in cell. Okay, they often cover the entire cell or the entire exposed surface of a cell while they are present on oftenly on a cell. Clear? And they show the sweeping or the roving motion while they show the undulatory motion. So the basic differences between the cilia and flagella. Clear? Now here we are moving toward the next cell component, that is called as the centrosomes and centriole. Okay, this centrosome and centriole is the basic characteristic feature of the animal cell. It is absent in the plant cell, right? One more thing, this is just considered as the cell division apparatus because it is participate in the cell division process, especially the mitosis and meiosis in the animal cell. Clear? Here you can see that a vacuolar structure is there inside it. The perpendicularly arranged, this centriole is present there, which is basically having the peripheral fibrils in the form of the triplets. Okay, and these are basically made up of the tubulin protein. Okay, and adjacents are arranged somewhat curved because here you can see that this ABC, ABC, ABC. So this A is attracted by C and C is attracted by A. So they are slightly tilted and twisted, which is responsible to give them this peripheral specific structure. Okay, otherwise you can see that if you observe then they are perpendicularly, then they give this some hollow cylindrical structure, and this way they are supporting in nature, they perpendicular on each other, and at the time of the cell division they go on the two opposite direction and produces spindle fibers, which is responsible to separate the chromatids or the chromosome from each other, and they are responsible for the movement, and they are also present where in the cilia and flagella, which is helpful for their locomotion or the movement. Clear? Now then here we are going to discuss the one most important cell organelle, that is the nucleus. Okay, the name of the nucleus is derived from the nuclein, which is the center, not only because of the position but also of its activity. It is considered as the master controller of the cell. Clear? It was first discovered by the Robert Brown in 1831 and later it was named by the Fleming. Okay, this nucleus is the largest cell organelle, right? And it is basically present in eukaryotic cell. In prokaryotic cell, only genetic material is present because of the absence of the nuclear membrane. It is termed as the nucleoid. Okay, now when we talk about the ultrastructure, then here you can see that okay, some cells are also anucleated too. As you know that exceptions are there. Likewise, the human RBC, it is lacking nucleus. Okay, so here in this ultrastructure you can see that it is covered by the double membranous structure, which is called as nuclear membrane. Okay, in this nuclear membrane you can find these pores are there, which is called as nuclear pore, which is responsible for the transfer of the messenger RNA from nucleus to the cytoplasm, as well as this outer surface of this nuclear membrane is sometimes extended to form the endoplasmic reticulum, which is responsible for the transportation of material. Clear? In this one, this fluid like feature plasma structure is there, which is considered the nucleoplasm. This one is consisting the different enzymes, different enzymes, especially DNA and RNA polymerase, which is responsible for elongation and the duplication and multiplication of the DNA. Right? Now here you can see that a spherical structure is present, which is called as nucleolus. Okay, it is naked in nature. It is not covered by the nuclear membrane. Okay, and if you observe that, then it is responsible for, it is also termed as NOR, nucleolar organizer region, where the formation of the mRNA is performed actually. Okay, and later on it is transferred. Then these chromatid fibers are there, which is made up of chromatin. This chromatin is divided into two parts, euchromatin and heterochromatin. Actually, when we stain them, then one who take the light color is called as euchromatin, and who take dark color is called as the heterochromatin, which is responsible for, out of this one thing, which is responsible for that hereditary transportation. If you observe that, then it stores the information for the which one thing, that is the control of the cellular activities. It controls the different synthetic structural proteins. It also stores the genetic information for the development, reproduction and the behavior Okay, it also induces the genetic variations too. Clear? So this is all about the nucleus. And now here we are going to discuss the one of the most important component of the nucleus, that is called as the chromosome. As you know that the word derived from the chroma and soma. Chroma means colored and soma means the body. First time when nucleus was stained by the scientists, then the structure which stained itself with that dye is called as chromosome. Okay, and this chromosome is basically made up of chromatids, okay, which is composed of DNA. As you know that the specific number of the chromosome is the quality and the characteristic feature of a species. Just like in our body, in a single cell diploid cell, 46 chromosomes are present in the form of 23 pairs. Okay, and this chromosome is composed of the primary construction and centromere and also this one thing. Okay, and on the basis of the position of the centromere, these chromosomes are basically divided into metacentric, submetacentric, acrocentric and telocentric. Okay, if you observe this diagram, then the telocentric, when this centromere is present at the end of one side and having only one arm, then it is called as telocentric. Second is the acrocentric, when this present centromere is present at the chromosomal at the peripheral end and one side is very negligible or very short, then it is called as acrocentric. Okay, and when it forms the L-shaped structure, it is slightly shifted from center to the peripheral side, then it is called as submetacentric. And when it is exactly present in the center of the chromosome, okay, and it forms this V-shaped structure, that is called as metacentric in nature. Okay, if you observe the functions of the chromosome, that is basically responsible for control the cellular differentiation. It is responsible for the transfer of the hereditary information from one generation to next generation. Okay, it forms a link between the offspring and the parents. You know that whatever the character which received by the offspring, it is because of the parental chromosome, and this introduced the variations to the process of crossing over. You know that at the time of the meiotic division, first reductional division, a process of crossing over is there where the genetic material get exchanged. Okay, and then the control cell metabolism. Okay, so this is all about today's discussion. Okay, I just want to tell you that please prepare the proper notes on this different cell component and the ultra structure of this one thing. Okay, because it is important from your examination point of view as well as for the next competitive exams too. So till that, wish you all the very best, do well and keep studying. Thank you.